Texas. I'd like to welcome everyone to today's GlomCon. Today we have our series with NephCure, Advances in APOL1 Therapeutics is the title. And just a reminder that our NephCure is an organization whose mission is to accelerate research for effective treatments for rare forms of nephrotic syndrome and provide education and support to those and improve the life who have protein spilling kidney diseases. And one of the resources they have on their website that we like to share with you is their Kidney Health Gateway. And this gateway has not only for physicians, researchers, family members, or patients, you can find information about ongoing clinical trials and specialists to better help serve our patients and advance what we know about kidney disease. And so, as I said, today's focus is on APOL1 Therapeutics. Our speaker today is Dr. Ogo Egbuna. He is the Senior Medical Director of Clinical Development for Vertex Pharmaceuticals' APOL1 mediated FSGS program, which aims to develop a treatment for underlying causes of FSGS. He completed his medical school at the University of Nigeria and his residency in internal medicine at Mount Sinai. He then did a clinical nephrology fellowship at the University of Rochester and a transplant fellowship at Beth Israel Deaconess Medical Center. Dr. Egbuna then completed a master's in clinical investigation from Harvard MIT School of Health Sciences and Technology and was on faculty teaching residents and caring for patients and conducting NIH-funded research at Harvard Medical School. Prior to his work at Vertex, Dr. Egbuna worked at Amgen as a clinical research medical director for both early and late phase clinical development programs. During that time, he served as an assistant clinical professor at the David Geffen School of Medicine at UCLA. He's the recipient of several awards and speaking invitations, and volunteers in his free time working for Kidney Solutions, a kidney disease and dialysis center in Laos, Nigeria, where he helps with standardization and implementation of policy, clinical procedures, as well as the use of modern technologies for dialysis and electronic medical records. Our two panelists today are Dr. David Friedman and Dr. Opeyemi Olabisi. Dr. David Friedman is a nephrologist at Beth Israel Deaconess Medical Center and an associate professor at Harvard Medical School. Dr. Friedman completed his medical school at Yale and his clinical training in nephrology at Beth Israel Deaconess. He studies the genetics of kidney diseases. His two main areas of interest are kidney disease in African Americans and the kidney disease epidemic in Central America known as Mesoamerican nephropathy. He was the co-lead author of the science report in 2010 that identified the APOL1 high-risk variants and has since co-authored more than 35 publications related to APOL1. His laboratory uses population genetics together with mouse models and cell biology to find genetic variants that predispose to CKD and understand the mechanisms of injury. Dr. Friedman's relevant disclosures are he's the co-inventor of patents related to APOL1, an equity holder in APOL1 Bio, and a consulting and research funding from Vertex. And our other panelist, Dr. Opeyemi Olabisi, is an assistant professor in the Department of Medicine Division of Nephrology at Duke University. He completed his MD and PhD at Albert Einstein College of Medicine, completed an internal medicine residency at Mass General, and his nephrology fellowship at MGH and Brigham Women's Hospital. He then went on to do a postdoc fellowship in nephrology at Beth Israel Deaconess Medical Center under the mentorship of Martin Pollack. He has received several awards. He is funded by the Harold Amos Medical Faculty Development Award from the Robert Wood Johnson Foundation and is the 2019 recipient of the NIH Director's New Innovators Award. Dr. Olabisi utilizes patient stem cell derived podocytes and reprogrammed mammalian cell lines as tools for uncovering disease mechanisms of APOL1 nephropathy. The focus of his research is to delineate the mechanisms by which kidney disease risk variants in APOL1 accelerate the progression of kidney disease in humans. So we are very excited to have all three of our speakers and panelists with us today. And with that, I will turn it over to Dr. Egbuna, and he will start the presentation.
All right, great. Just give me a second to put up what I have. I will say that it's an honor to be here amongst friends and colleagues. I know quite a number of you. Dave and I were actually fellows at the BI. Actually, at the time, I didn't realize all that he was doing. I was actually working with Ed Brown at the Brigham at the time, phenotyping mice for Martin Pollack at that time, I think he was working quite a bit also on the calcium sensing receptor. So that was some interesting work to sort of look back on. So let me just get started here. So we'll be talking about the Vertex small molecule therapeutic development program. And, I'm really excited to be kicking off this meeting today. Just in case you're not familiar with Vertex, Vertex is a global company that invests in scientific innovation to discover precision medicines for patients with serious diseases, such as cystic fibrosis, where Vertex has transformed the lives of patients with what was previously known to be a devastating illness. At Vertex, and it is one of the reasons why I decided to move here. Sorry, there's an echo here. I'm not sure why. So we don't look to make marginal progress. And I guess maybe if you're not speaking, maybe if you mute your line, I think that may help. All right, thank you. So so at Vertex, we don't look to make marginal progress. We take on problems where we think we can have transformative effect. And that's because we only pursue, approaches where there's strong evidence to address the underlying cause of disease and not a downstream process. So we're adopting very much the same approach which we've applied quite successfully to cystic fibrosis, to our emerging pipeline that includes sickle cell disease, alpha-1 antitrypsin deficiency, type 1 diabetes, and APOL1 mediated kidney disease, which is the subject of our conversation today. So to start our conversation, I'd like to, make take us, ground us on the reason why this is important, why all the fuss. And it is because the burden of kidney disease is quite large. Kidney disease is amongst the growing causes of mortality. And from this report, a bit dated, but still relevant, in 2013, 1 in 57 deaths worldwide was attributed to chronic kidney disease, and there has been a substantial increase in the number of deaths from kidney disease over the last 2 decades. The economic burden of kidney failure, I think, is quite substantial as well. And just looking at these figures here, you can see that as patients progress from less severe degrees of renal impairment to end-stage kidney failure, there's a substantial increase in cost. Much of that cost is driven by the excess in myocardial infarction, stroke, prolonged length of hospital stay, and infections. And I think a very important point to mention here is that 5% of the budget for the Centers for Medicare and Medicaid is spent on the less than 1% population of patients that have end-stage renal disease on dialysis or transplant. And a similar, substantial budget implication is seen in the UK with the National Health Service, where 1.3% of the budget is spent on, certainly less than 1% of the population. We've also noticed that there is a disparity amongst kidney patients. And whether you look at the US renal data system or the UK renal registry system, you'll see that patients that identify as Black have a higher cumulative incidence of kidney failure across the entire age spectrum. Now, there are lots of causes that I think we are well recognized to be the cause of this disparity. Some of it being attributed to access to healthcare, socioeconomic factors, but there certainly does appear to be a genetic basis for some of this disparity. And as Dia had mentioned, the paper in Science in 2010, which David co-authored, did bring out, or did explain quite nicely the association between tripanylated APOL1 variants and kidney disease in African Americans. Just looking at this slide, you can see that if you look to the far right, IgA nephropathy and diabetic kidney disease, for instance, very prevalent in the patient population, do not appear to have any association with the APOL1 genetic risk variants. But when you look at diseases such as hypertension-associated ESRD or focal segmental sclerosis or even HIVAN, you see that the odds ratio associated with these genetic risk variants is quite high, and that's an unusually large effect size for what are common variants of the APOL1 gene, which, as we all know, is the protection that has evolved over time to resistant forms of human African trypanosomiasis. So in addition to increased risk for disease, the APOL1 risk variants are also associated with rapidly progressive disease. And whether we look at focal segmental sclerosis or hypertensive kidney disease, you can see that those with 2 copies of the APOL1 risk variants, progress at a rate that's substantially higher. And just, taking your attention to this figure on the left-hand side, 50% of patients have reached a terminal endpoint of needing dialysis or transplant or death within 5 years when they have 2 risk alleles and a diagnosis of FSGS, while those with 0 or 1 risk variant have a much longer survival in the order of, with a median survival of about 13 years. So since the identification of the APOL1 gene and its relationship to progressive kidney disease in patients of recent African ancestry, there's been, a lot of investigation into where the APOL1, where these APOL1 genetic variants exert their effect. And I think most people would agree that the podocyte is the cellular target of APOL1, and that inflammation, be it in the form of infections or hypertensive renal damage, triggers the progressive disease that is seen, and that has driven the associations that have been identified with APOL1 genetic variants and progressive kidney disease. And it's this toxic gain of function of the APOL1 genetic variants that leads to cell death and disruption of the glomerular filter. The therapeutic hypothesis that Vertex and many other, and a couple of other sponsors have been pursuing is that inhibition of APOL1 activity would impact or inhibit the progressive disruption of the glomerular filter that is seen. And Vertex, as part of its small molecule inhibitor program, has taken a number of molecules with a lead molecule called VX147 into the clinic. But before then, there was a whole extensive effort using preclinical, in vitro, and in vivo assays to identify highly potent, very specific inhibitors of APOL1. And these have been successfully taken into healthy volunteers, where VX147 has been shown to be safe and well tolerated. And since the completion of those first in human studies, Vertex has gone on to initiate a phase 2 proof of concept study. This study is ongoing, and the primary objective is to evaluate the ability of VX147 to reduce proteinuria in patients with APOL1 mediated FSGS. The sample size is a small study, open label, single arm trial with a primary endpoint of % change in baseline and urinary protein to creatinine ratio at 13 weeks, and a secondary endpoint to evaluate safety, tolerability, and the pharmacokinetics of VX147. As you can see in the schema in the middle, there's a screening period during which eligibility of potential patient subjects are determined. And then there's a 2 parts portion subsequently, with part A, 13 weeks on label, on treatment, and then an optional off treatment observation period. The key eligibility criteria are for subjects that would qualify for this study involves adults aged 18 to 65 with biopsy-proven FSGS and 2 APOL1 high-risk genetic variants. Genotyping, which we recognize is not commonly done, is offered as part of the screening process to determine patient eligibility. We are excluding patients with known secondary causes of FSGS, and we're excluding patients with the TIP variant. We are in a pandemic, and I'm sure most of you have read papers and heard conversations about COVID-19 related nephropathy. Patients with a previous history of COVID-19 infection are not excluded, but patients with active COVID are excluded. As would be expected, we're looking at patients with proteinuria, and we're allowing low-dose steroids, tacrolimus, and mycophenolate as part of background therapy. And I'd just like to say that VX-147 is not approved by any health authority. It's in the therapeutic development process, and as such, the safety and effectiveness has not been established. So this is my last slide, and I just like to say that if you need to know more about this Vertex clinical trial in FSGS, feel free to email me, or you could direct your patients to our website at fsgs.apol1studies.com. And at this website, patients could see if they qualify, they could learn more about the study and FSGS as a disease, and they could even find the nearest clinical trial site to them. So with this, I will stop and say thank you for your attention. It's been a real joy for me to be working on this program, especially seeing all the progress that has been made in a relatively short time since that seminal paper in 2010 in Science. So with that, I'll stop and say and hand back to Dia. Thank you.
Thank you, Dr. Egbuna. To the audience, we started with kind of an overview of a drug that's in development, and we're kind of going to take just a little bit of step back for the discussion and answer some of the questions that have been coming up kind of in the chat. And to do that, I kind of want to give you kind of a roadmap of how we'll try to go through this so we can hit on a lot of these points that are already being asked, and then relate them back to your ongoing research and drug development. And so the things we wanted to kind of touch on is I'm going to ask, first Dr. Friedman and then Dr. Olabisi to go over, Dr. Friedman to talk just a little bit briefly on the genetic lineage and of APOL1, because I think that's important to have a background to understand why. Then Dr. Olabisi can comment on a little bit on his research and why it's so hard to make these animal models to understand the function of APOL1. And then from there, we'll kind of talk about, what we know about the mutations and what we're trying to fix or inhibit in them, talk a little bit about the clinical presentation and spectrum of disease, go on to therapies, and then finish up with, who we should be genotyping and such. So just to kind of give everyone a little bit of a roadmap of how we want to kind of things we're going to touch on and highlight during this discussion. So we'll start with Dr. Friedman, and if you could just comment a little bit on the, some of the work you've done and some of the genetic lineage and how it hasn't affected all mammals. And so we can then lead into Dr. Olabisi to talk a little bit about the challenges in the lab of understanding the function of APOL1.
Sure, thanks, Dia. So APOL1 is actually a very recent gene in mammalian evolution. It probably only came about from gene duplication from some very close neighbors about 30 million years ago. And what that means is that only primates have the APOL1 gene in their genome. And in fact, not even all primates do. It's mostly restricted to old world primates like gorillas, baboons, mangabes, mandrills, and humans. So as Opie will talk about, it's been a challenge to come up with animal models of disease. The risk variants themselves, which we have nicknamed G1 and G2, are also very recent evolutionary adaptations. We're guessing that they're probably in the range of 5,000 to 10,000 years old, and they originated in Africa in the Trypanosome belt, long after the out of Africa expansion that populated the rest of the world. So when people left Africa, somewhere between probably 50 and 100,000 years ago, the high-risk variants didn't exist. So people for the most part, people in Asia and Europe of Asian and European ancestry don't have the APOL1 high-risk variants. In West Africa, the variants are particularly common, and now in the United States, they, because of the strong influence of West African ancestry in the Black population, they're quite common. So one of the risk alleles, G1, is present in something like 23% of all alleles, and G2 is present in about 15% of alleles. We think maybe 13% of all Black individuals in the United States have the high-risk genotype. And by the high-risk genotype, what I really mean is 2 risk alleles. It turns out you need 1 risk allele inherited from your mom and 1 from your dad to get the high-risk genotype. Surprisingly, people with only 1 risk allele don't really have much of an increased risk of kidney failure, maybe a very small increased risk, but with 2 risk alleles, the odds of disease go up sometimes as much as an order of magnitude or more. Dia, would that be a good place to take a break?
Yeah, no, I think that's perfect. And then if Dr. Olabisi, if you could just comment a little on why it's been so challenging to study these because of kind of their genetic background and not having a lot of animal models to go off of, and how you've kind of overcome a little bit of that to get to where we are now to better understand it.
All right, thank you. Can you hear me?
Yes.
All right. So I'm very grateful to be invited to speak here. I see a lot of my mentors and teachers, and also I see Martin somewhere on the forum. So I'm humbled to be addressing this August forum. So as David has laid it out, I think the genetics led the way. The genetic epidemiology pointed the direction that this polymorphism is important. Then the question that followed that closely was, is this variant, this APOL1 variant, is it causal or is it just a risk factor? Those were the initial questions that people were interested in, right? That people that have this variant, when you have this variant, is it what's causing the diseases or is it just a risk factor? So the best way to approach that is to take a reductionist approach and say, what if we express this in model system, experimental model system? Now, as David had said correctly, a lot of our experimental models, mice, even zebrafish and all that, they do not have a APOL1 gene, at least as in case of this experimental model. So if you want to study them, you have to sort of make a transgenic model. And there have been several approaches that have been taken. So first, in Dr. Pollack's lab, I remember when I was there, training there, we put this in zebrafish kidney to see if you put APOL1, either the reference variant, G0, or the risk variant, G1 or G2, what do you see in the kidney? Now, it was actually interesting because we saw that the ones that express G1 or G2, which are disease associated, you see evidence of injury in the glomeruli, but we did not see florid proteinuria. But the ones that express G0, you do not see injury. So that was kind of interesting model then. Part of the issue might be that the zebrafish has a different physiology. They regenerate their pronephros. Since then, there have been animal models that are actually probably more physiologically relevant. The first one was from the Brueggemann's John Sadler's group, where they made a transgenic APOL1 mice, where the APOL1 expression was driven by the nephrin promoter. So it's heavily driven in the kidney podocyte. What they saw there was interesting. That was the first model. And they saw that there was a reduction in the podocyte number, but there was no FSGS or frank kidney disease. Now they saw that only in the I believe in the G1, they made the G1, not the G2, but they didn't see it in the G0. Subsequent lab or other labs have since made other animal models. So followed by Kathleen Zustak lab that made a tetracycline inducible APOL1 in the podocyte. And in that model, you see a florid proteinuria. When you do kidney histopathology, you see FSGS that mimics what you see in human, but you only saw that in the risk variant, but not in the G0. Other group, the AstraZeneca group, the Jeffrey Cobbs group, Martin Pollock group, subsequent mouse model literally have affirmed that APOL1 risk variant is actually causal. When you express the G1 or the G2, it drives kidney disease, especially in the podocyte. Cell culture model have also supported this. When you express the G1 or G2, you see injury. When you express G0, not so much. So all these experimental models have essentially, I would say, strongly answered the question that this variant is actually causal of injury. The question that remains unanswered, which will be difficult to answer in animal model, is that why is it that some people carry this mutation but never develop disease? What is the basis of the incomplete penetrance? I think this would be a good place to stop also.
Yes, excellent. And I think based on the background that you've given and where we are with understanding it better, we know where it is on the gene, what's encoding. We know it's inherited in an autosomal recessive form. And so when you think of autosomal recessive, you think of loss of function, but that may not be the case, correct? And so if you guys could comment a little bit on whether, what we know and where we are and whether it's a gain or loss of function, because I think that has implications as we go down the line in developing therapies and what we're actually trying to achieve in those therapies.
So I can maybe take a first hack at it, or David? Okay, so when you think of a gain of function mutation, just to define the terms, we think of a mutation that confers a activity on a protein. When you think of a loss of function, you think of a mutation that reduces or abolishes a protein function. So if we are thinking of APOL1, the first question is, if this is a gain of function mutation, do the G1 and G2, have they acquired a new function that is absent in the G0, right? But I would say that and we are thinking of this in context of kidney and kidney disease, so broadly speaking, because in the context of infectious disease, if you lose APOL1, you acquire increased risk of infection. There's one reported case of somebody who is null, who doesn't have the APOL1, express the APOL1 gene in India. What was seen in that person was increased risk of trypanosomiasis, but the person seems to have normal functioning kidney disease, normal functioning kidney. That suggests that the APOL1 gene or the gene product may not be necessary for the normal development of the kidney. Now, one might say, maybe there might be a function under stressful condition, or that we don't know that for sure. But in terms of normal functioning or normal development of the kidney, absence of APOL1 does not seem to be necessary for that. That argues against a loss of function, meaning that when you lose this APOL1, the kidney continues to function normally. Stronger evidence comes from experimental model. So if we stick the APOL1, either the G0, the G1, or the G2 in a mammalian cell, what do we see? At least in Martin's group, when I were there and also confirmed by others, we see increased loss of cation, increased flux of cations. Now, for people in the audience that may not be following this on a daily basis, APOL1 also acts as a cation transporter. It moves cation, it moves potassium down its gradient, it moves sodium down its gradient. G0 does that, but the G1 and G2 seems to do it more so when we express them in the mammalian cell. So when we look at toxic function, if I zoom out, when we look at toxicity in the cell, the G1 and G2 seems to have a gain of toxic function. And in fact, a recent study that we just published shows that this gain of toxic function also seems to be dose dependent. So if I can zoom out, I said the experimental model strongly suggests that the toxicity of G1 and G2 are actually a gain of function, not loss of function mutation.
Opie laid that out beautifully. I might just add a couple things. First of all, like we talked about, you know, all the other mammals don't have the APOL1 gene. Even the chimpanzee, our closest relative, turns out not to have the APOL1 gene in the genome. And it would be very surprising if in human evolution, we suddenly found a need to have this gene that the chimpanzees and all the other animals don't, unless it's very situation specific, or as Opie pointed out, in response to some stressful environmental conditions. And again, the individual identified in India with two completely different null mutations, that person has normal blood pressure, no proteinuria, totally normal kidney function. Again, it would be a big surprise if this was truly an essential gene for kidney function. We really think it's not a kidney gene so much as an innate immunity gene, which has a role in the immune system, not in supporting the function that we generally attribute to the kidney. So, you know, I think one may think of this as, let's suppose that APOL1 is a toxic molecule. It's possible that the C-terminus, where both these variants are, might be an inhibitory part of the molecule. And in that case, just one of many possible examples is the variants in some way impair the ability of that inhibitory part of the molecule to prevent the toxicity. So even though they may in some way impair the part of the domain specific function of APOL1, that in total the APOL1 activity in terms of toxicity is much increased. And we still can't quite understand why some people get the disease and others don't. And we really are looking for the triggers or the other genetic modifiers, which will help us figure out why some people do go on to get disease.
Thank you. And then I think along those lines, so we know that APOL1 circulates, as you said, it's maybe part of the immune system, immune response, but there's also localization in the kidneys. And so you can measure circulating APOL1 levels be high, but maybe not have the kidney damage. Can you kind of comment on, do you think it's a problem of kidney localization? Along those lines, in the kidney, we see it in the podocyte, but it's also in the endothelial cells, correct? And so why is that? Maybe, and this is, I think, our second part of the question, which there's some comments on as well, is why do we see this spectrum of, FSGS to hypertensive nephropathy there? So.
Should I start this one, Opie?
Yeah.
Okay. So, because we don't have great animal models and our cell models have some deficiencies as well, we really like to try to learn from natural experiments and clinical observation how APOL1 behaves in humans and its mechanisms. And I think, as Dia pointed out, circulating levels in the blood of APOL1 are high, but it's also expressed by the kidney, particularly under settings of inflammation. And one way we've been able to learn about the difference between those two possible, culprits is to look at the transplant literature and to observe what happens when a transplanted kidney from a person with a high or a low risk APOL1 genotype the graft is placed in someone with a high or a low risk genotype. And to briefly summarize, it turns out that the risk travels with the donor kidney, the donor genotype. So if you put a high risk kidney into a person, it will fail. It will have a faster time to failure, whereas a low risk kidney doesn't appear to behave much differently than a kidney not from a Black individual. So what we think is that it's the kidney expressed version of APOL1 that is the toxic entity, and the circulating form probably less so or not at all, though I don't think the final word has been written on this topic.
Yeah, and if also to add to that, why so the spectrum, and indeed what we saw is, at least what clinically evident is that when you look at the APOL1 disease, seems to be a spectrum of disease. The review article by David and Martin that shows the odds ratio, you can see that you go from HIVAN, things like HIVAN that in Africa, South Africa, the odds ratio is as high as like 89. In the US, like 29. It's really incredible odds ratio. All the way down to things like lupus podocytopathy. But I would say that one way of looking at this spectrum is to look at it in terms of target cell in the kidney. It seems to be that podocyte seems to feature quite a lot in many of these diseases, as attended also by proteinuria. But by no means should we think of this as a exclusively podocyte disease because of the hypertension ESRD. In fact, that might end up to be the largest group because of the impact of, so I would say that we don't understand the mechanism of how APOL1, for example, causes hypertensive nephrosclerosis. There's more attention focused on how it causes podocytopathy. Even that we don't know yet. So I think as we know more about cellular mechanism, we probably will understand why different cellular compartments are differentially vulnerable to the higher effect of the toxic APOL1. I think there's an insight from Kathleen Zustek's mice that she published in Nature Medicine way back. When she overexpresses APOL1 in a podocyte exclusively versus expressing APOL1 exclusively in kidney tubular epithelial cell, she saw injury to podocyte and FSGS and proteinuria in the podocyte, but she did not see any evidence of injury in the epithelial cell. Protecting the epithelial cell and what makes podocyte more vulnerable. We have less information about the endothelial cell. So cellular mechanism will probably help us to understand this disease spectrum.
Thank you. So then Dr. Egbuna, you showed us your molecule that's in development that inhibits the APOL1. So can you talk a little bit more about like maybe why that was the target chosen when we're talking whether it's inhibitory and what you have some data on healthy volunteers. Has that shed any light into what we've just kind of discussed?
Yes. So can you hear me?
Yes.
Okay. So the working assumption here, based on what I had mentioned, our suite of preclinical in vitro assays, is that that APOL1 genetic variants have toxic gain of function, and be it cation flux or trypanosomicidal activity, the APOL1, like VX147, has been shown to abrogate those functions of the genetic risk variants. So that's the working assumption that we're working with going forward, with the understanding that the podocyte is an important target, but also, like Opie had mentioned, the endothelial cell is potentially also implicated in the manifestations of disease.
Okay, thank you. And then along those lines, what clinical presentations of disease, you showed us some of your inclusion criteria, but in general, what do you think that our current understanding will lead us to? Which patient populations are we going to be directing therapies to, or who may benefit more from these therapies based on what we know about the APOL1 thus far?
Well, I think it probably goes without saying that treatments would be expected for use in patients that have manifest disease, because as was mentioned earlier, it's the action. We don't know why there are a number of patients with 2 risk alleles but do not have any evidence of kidney disease. So treatment would be limited to those with 2 risk alleles or the high-risk genotype and evidence of kidney disease. I think the consequences of disruption of glomerular function driven by protein spilling like edema, the hypercoagulable state, secondary hyperaldosteronism, are what is expected with precision therapies to be impacted by therapies targeting the toxic gain of function activity. I think there was one other aspect to the question that you asked. Please remind me. I think I might have forgotten that.
Oh, no, I think it was essentially what would be, what disease process and essentially what phenotypes of this disease, right, or this risk would be most amenable to treatment, or who are we looking to target? And then, from Dr. Friedman and Dr. Olabisi too, of, where do you think the future kind of treatment is going, and which clinical phenotypes would we potentially expect to respond if we have treatment to stop this process?
Yeah, so with regards to which patients, I think the evidence is most compelling for those with 2 risk variants. The evidence about the role that patients with 1 risk variant have on the progression of disease is not as robust as it is for those that have 2 risk variants. So I think those with manifest kidney disease and 2 risk variants are those that therapies, at least for starters, would be directed to. And perhaps as evidence emerges on the circumstances under which those with perhaps just 1 variant might be at risk, we could then explore, the value of treating such patients. But I don't think the evidence supports is as strong for those with just 1 risk variant.
And then, and when you guys comment on that, can you talk a little bit about the kind of second hit phenomenon too, where you may have 1 and what predisposes potentially injury in that regard?
Yeah, Opie, you want to take this one?
Sure, I'll be happy to. So to also put a little finer point on, so who should be, what should be the indication for the treatment? So if you look in population-wide screening, people who have not developed advanced stage kidney disease, proteinuria seems to be a very sensitive predictor, or is more strongly associated, if you will, with the high-risk APOL1, more so than low GFR initially. So I would say people that have proteinuric kidney disease that have the 2 risk allele will be, at least in the initial sense, that should be the focus. Now, what about people that have low GFR without proteinuria? I don't think we know enough about those yet. But I think there should be a lot of focus on people, people who have high-risk APOL1 genotype, who have proteinuria of all range. Now, we do focus a lot on nephrotic range proteinuria, but actually, if you look closely, a lot of patients with APOL1 related kidney disease have sub-nephrotic range proteinuria. And so we should not ignore that fact. So proteinuric kidney disease, 2 risk allele. What about people that have hypertensive nephrosclerosis? As Barry Friedman from Wake Forest has pointed out, that the attribution of CKD in probably a lot of African Americans may be attributable to high-risk APOL1 genotype. So I think as we learn more, hypertensive CKD in the context of high-risk APOL1 will be probably one of the largest indication of treatment moving forward. You asked a question that evades me just now that you wanted me to comment on again.
Oh, there was a question about the second hit.
Second hit, thank you. So, so one of the theory is the 2-hit hypothesis, if you will. That why is it that some people have the high-risk genotype but never develop kidney disease? One of the common prevailing theory presently is that the high-risk genotype is the first hit, but that a second hit is needed to actually, shall I say, trigger disease. And a few of those have been identified. The first one are interferons, where the group at Columbia had a cohort, if you will, a case series of African Americans that were found, or actually initially they were just a cohort of patients that developed collapsing FSGS in the context of receiving interferon, therapeutic interferon, alpha, beta, gamma. And when the APOL1 story came forth, David and Martin were able to partner with the Columbia group, and they genotyped a lot of those patients. And it turns out that majority of them that were able to genotype actually were high-risk APOL1 genotype carrier. So that was the first hint that there's a convergence between exposure to interferon and the unmasking of the risk of podocytopathy. David's lab went on to show that if you expose the cells, podocyte or endothelial cells, to interferon, interferon actually causes the expression of APOL1. Now, we've seen, and this has held out very strongly. So interferons or environmental triggers of interferon, high level of interferon, is emerging or are emerging to be triggers or second hit factor of APOL1. HIV is probably the strongest known driver or second hit. About 50% of African Americans that have 2 risk variant APOL1, when they have uncontrolled HIV, develop HIV. That's really remarkable. So HIV, and when you treat the HIV with the ART therapy, that risk pretty much disappears. So HIV is a very potent second hit. The COVID pandemic is also unmasking or emerging as another hit. So COVID is strongly associated with collapsing FSGS among African Americans. There have been about 40 cases reported both in the US and from France. Whether this is acting through interferon or whether this is acting through viral exposure to the kidney, though there's not a strong evidence that these viruses are actually infecting the kidney directly, mediating the injury. But there's you can think an axis of virus. We know that HIV does it. We know that COVID does it. And there have been cases in transplants where CMV and EBV have also been indicated to a lesser degree. And in addition to that, the second hit that I mentioned is interferon. But I would say that there's probably other second hits that we have not yet uncovered. So we have to keep an open mind about that.
I might just add to that. Yeah, I think Opie put that extremely well. In the more aggressive forms of APOL1 nephropathy on that spectrum that Ogo showed, it does seem like viruses and the innate immune response to the virus are likely to be the major driving forces. And they're not just the immune, not just the pathogen activated side of the immune system, but also things like, like lupus is a very high interferon state. Other rare diseases like HLH is a real overwhelming autoimmunity syndrome, also appears to be able to activate APOL1 kidney disease. And I think the real challenge is going to be to figure out what causes or acts as the trigger in the more common forms of APOL1 nephropathy that used to be called hypertensive CKD or hypertensive ESRD. But I think there's a good bet that there are just less aggressive forms of inflammation, maybe related to diet or other environmental factors that don't cause explosive upregulation of the APOL1 protein, but maybe more chronic subtle upregulation of the APOL1 protein that lead to a less dramatic phenotype.
Yeah, and to that point, in support of what David just said about maybe more indolent disease happening that we are not capturing. There's a recent paper from John Cedar, first author Dorothy Shen, where they look at living donors who basically donated kidney, and they were able to actually count, I think they did podocyte density. And what they showed was that there's a correlation between having a high risk APOL1 and having a reduced number of podocytes. Now, it's a small number, but you have studies like this that shows that there might be indolent, undetected loss of podocyte over time that is happening in the kidney of some patients. And I'll say last point on this, in terms of understanding the interaction of the second hit. I mentioned that interferon has been identified as a potential second hit. And this point probably highlights maybe the importance of genotyping. I don't mean to jumpstart the question, where in the context of COVID, deficiency in interferon signaling has been indicated to be important mechanism of COVID, severe COVID. So much so that some people have now advocated for using interferon treatment as a treatment for COVID. Now imagine if you are treating an African American that have 2 risk variant APOL1, you think you are treating the COVID, but you're applying them with a high dose interferon. There may be a lot of unintended side effects. So I think this, the need for genotyping is underscored by the drive to use interferon in therapeutic aspect of COVID. And I don't think we should jump into it without thinking about that aspect.
No, and before we move on to the big question that's about genotyping, there's a question back what Dr. Friedman mentioned about the spectrum that Dr. Glassick had asked. And basically the question was, you know, in the ASK trial biopsy sub-study, it was troubling that many of the lesions of FSGS were uncommon among the African Americans with hypertension, despite the expected prevalence of APOL1 risk alleles in the cohort. And most of the biopsies showed focal global glomerulosclerosis, not focal segmental glomerulosclerosis. Do you guys have any comments on or explanations for that? Or is it possibly part of the spectrum of this APOL1 nephropathy?
I think this might be related to how we choose which of our patients to biopsy. I tend to think that Black patients get biopsied later and often not at all when a white patient may be biopsied with the same clinical presentation. And I think that that is probably why more global sclerosis is discovered or identified by the biopsy. And it is totally possible that much earlier in the disease course, you might find a higher fraction of people who have FSGS. FSGS will eventually go through the final common pathway that all kidney failure does, which is global sclerosis.
Thank you. And then another question he had was, are there differences seen in the course of hypertension or HIV disease in African Blacks indigenous to Nigeria compared to Ethiopia?
So I think we have more data from the US with USRDS. There's no equivalence of USRDS in Africa, but there are in some case reports. For example, there was a recent case report from Ghana, which is pretty close to Nigeria in terms of the frequency of APOL1 and disease. So they looked at patients that have lupus podocytopathy. And it turns out that the odds ratio of lupus nephropathy or podocytopathy among people that have high-risk variant is 14, which is significant. In fact, it's like double what we see among African Americans. So first, the odds ratio is higher. And the progression to ESRD, as well as mortality, is, of course, in Ghana, just like in Nigeria, dialysis is not paid for by the government. People pay out of pocket. So if you have end-stage kidney disease in the resource challenged environment, it's essentially a death sentence. So it's almost uniformly deadly once it advanced to end-stage kidney disease. But what we saw in that paper was that the odds ratio seems to be higher, at least for APOL1 associated lupus nephropathy. And the time to end stage is also significantly faster. I don't have the number, but I think we need more data from Africa. The H3 Africa is looking into this, and I think with time, we'll be able to see a difference. About Ethiopia, though, for Hyvan, that was very illuminating because I'm trying to attribute the group. David will probably remember the name. When they look at Hyvan and Ethiopia, actually, there's really, because Ethiopians don't have high-risk APOL1 genotypes, the incidence of Hyvan, I believe, was significantly lesser in that population. In fact, it's one of the natural epidemiological data that supports the fact that APOL1 is indeed a huge driver of Hyvan in the African American community and in sub-Sahara West Africa, most especially.
Thank you. And in the remaining time that we have, I think one of the big questions and that questions that have come up in the chat have to do with, one, availability of genotyping, two, who should we be genotyping? And then how are we educating patients on, this genetic screening and what it means to them?
Okay, I can take a stab at that. I think genotyping for APOL1 genetic variants in patients with kidney disease certainly has not yet become a common recommendation for management, but practice certainly is changing. As for any disease, I think, and as has been mentioned, just a few moments ago, genotyping serves multiple purposes. It guides decisions for treatment where specific treatment exists, and where specific treatment does not exist, it may help influence decisions not to use certain therapies that are unlikely to hold a valuable benefit-risk profile. Genotyping also guides decisions on important life decisions, and such as the decision to donate a kidney or to proceed with pregnancy in patients with CKD and a past history of eclamptic disorders. I mean, I'm quite sure that many of us on the call today are aware of, women, with a past history of CKD having exaggerated eclamptic response and ending up on dialysis. So, there are lots of reasons, even in the absence of a specific treatment, to genotype. I think the opportunities to genotype are increasing. Invitae offers, this service. Natera offers this as well. In the context of our trial, we're really offering it as part of an assessment to be assessment of eligibility for the trial, and not as part of general practice, since it's not a recommendation per se. So maybe I'll just stop there and offer Opie and David an opportunity to weigh in on this.
Yeah, I can go next. I think in terms of indication for genotyping, so maybe we should say, why should we genotype, right? So one indication is to encourage targeted therapy. APOL1 does not cause all FSGS, but we think, or let me even put it more broadly, podocytopathy or kidney disease. But we want to identify the kidney disease or patients that have kidney disease caused by high-risk APOL1 genotype. So if I have a patient with FSGS or FGGS or any of the spectrum of podocytopathy that have African ancestry history, knowing that that person have high-risk genotype may help my thinking that this may be somebody that is eligible potentially for treatment for APOL1. Now, in the past, we don't have treatment. We still don't have treatment, but we have a group like Vertex working on one. When treatment becomes available, I think one of the immediate application of genotyping will be to have targeted therapy for individuals that have APOL1 nephropathy. So I think that's the lowest hanging fruit. The next rung is to actually identify people who are potentially at risk. And this area is more fraught with uncertainty. Because if we know that the frequency of high-risk genotype in Black Americans, as David mentioned, is 13%. But available back of the envelope calculation shows that only about 20% of the high-risk carriers develop disease. What we lack currently is the ability to differentiate those 20% that are likely to develop kidney disease from APOL1 in their lifetime from people that will remain healthy. So if you do a population-wide screening, you will be able to identify the 13% but that's where you will be stuck. Currently, we don't have a way to stratify people who are high-risk carriers who will develop disease. These are people who are currently healthy, by the way, who will develop disease versus people who will not. I think as we learn more, as we can stratify that population, it will make population-wide screening more, should I say, tenable. But on the converse, I mentioned the story of the COVID as we speak. If you're an African-American, if I'm an African-American in the hospital and somebody wants to treat me with interferon today for COVID, I will be worried. Because if I have high-risk APOL1 genotype, I probably don't want the interferon for my treatment for COVID because I will be at a higher risk of developing COVID-associated nephropathy associated with APOL1. So I think as we learn more about the basis of the incomplete penetrance, it will be more possible to do a population-wide screening. And I dare add one more point in terms of the issue of, because this has come up even in our academic setting, that is APOL1 a form of racialized medicine, right? I think this is actually the opposite. If you want to have a targeted, personalized medicine, this is one example of how you can achieve it. Instead of grouping a whole group into one basket, knowing the genotype of that patient allow you to provide a personalized care to that patient. So the use of APOL1 in both in clinical research, both in basic research, both in clinical medicine, is actually promoting personalized, individualized care rather than some form of racialized medicine. I think that's worth mentioning.
I'd just like to add one thing, which is that transplant nephrologists are already using APOL1 genotyping fairly widely. They're using it to risk stratify the kidney graft. And as many people in the audience probably know, there's a very large study going on called the Apollo study, interested in both the survival of grafts, but also the impact on the kidney donor. I think there's some debate about whether Black kidney donors should be genotyped for APOL1, but I think at least every potential donor should have the right to know whether they have that high-risk genotype and whether, and it's very important for the study to figure out what the impact is on their outcomes. Maybe just one other point is that a real barrier here is the outrageous price that genotyping centers are charging for this genotyping to be done, often $500 or more. This is a test that can be done for a few dollars. There's always a cost associated with making it a CLIA certified test, but we should be able to provide this test widely for practically nothing. And I think that's a really important thing to work toward.
Thank you. Well, I think that we're just back at the top of the hour. If there's any last minute points or questions from the audience too, please feel free to unmute. This is a very hopeful topic that we can find some answers and have therapeutics in the near future and have personalized medicine for our kidney patients, and then be able to educate and inform them too, as you said, Opey, about this and what it means to them. So with that, do you guys have any last comments or any questions from the audience?
Well, for me, I think it's been an honor to, be able to be part of this conversation and share a common knowledge about where this story started, and where we are right now. I think it's remarkable that in, the span of about 10 short years, we're actually talking about therapeutics and, broadening our knowledge about how to, more precisely treat patients rather than just bring out the usual cyclosporin, tacrolimus, and steroids and hitting everyone over the head with it. So, I'm really hopeful for the future, and look forward to what might become of the trials, not only that Vertex is doing, but others.
And a big thanks to the GlomCon team for helping bring this to wider attention. And it was a privilege to be able to talk about this topic with Opie and Ogo. They're real major leaders in the field, and really very hopeful about the prospects in the future.
Yeah, and I also echo the same thing that for this forum, GLOMCOM and Neptune bringing this, making this forum available. And also thank all the people within the field that, you know, David is extremely instrumental. It was, you know, it was my mentor with Martin Pollack when I was in Boston. And we need more people like you, David, and Martin, and others that are championing this, I would say personalized, if you will, understanding the basis of kidney disease in the Black community. And I think we need to do a better job, as we are currently in this forum, letting the community know that this is not racialized medicine. In fact, this is the direction we need to go to understand the biological basis of kidney disease. It's not the only cause of kidney disease, but it's something we need to pursue more. So thank you for making this available for discussion.
Well, thank you guys all for all of your input. Thanks for all of your work on this, and thanks for spending your Sunday with us and sharing your knowledge. And to everyone who participated, appreciate all of it. And everyone, be careful, and we'll see you guys next week. All right. Thank you.
All right. Bye.